Comments, you're good people. Don't send us not good.
Problem is I got.
But it requires like a 2 outside mentors, 2 years. They have to come to campus once a year. Mentor, and they only mentor.
Oh, you're actually outside.
Amazing. And you get your thinking about it. And we show them. Point out. And when an outside person, everybody's nonsense. And so if you have a established thinks that mentoring this person so he can have enough paper, or as I saw at UT Southwestern, gives that guy the day that says you analyze mine before you analyze your own. That doesn't happen because the minute you bring in stuff like it's very clear that this meeting is with this person. You know, this is about office. Doesn't all be the chair doesn't do so they write the report. They break it. Really cool. Yeah. And there's something, what is this saying that I've been an expert somebody was I can tell you all right.
See, I'm an expert today.
And but I can tell you
Yeah.
and then
Okay. All right. Hello. Long time no see.
Thanks for coming.
All right. You ready to get started?
We are.
Okay. So for some reason, Anna trusted me to stand in and do Good News Thursday today. So for those that aren't familiar with what we do at Seminar, we like to highlight some of the good work of our staff and our students before the wonderful presenters get started on their talks. So we only have one piece of good news today, but it's a big one. So congrats to Milad for passing his comprehensive.
Yay!
That's all.
All right. And I'm going to take 30 seconds to remind you that we had 2 lectures this semester funded by Dewey Shoddy. And let's just read this. This is a picture of Ron Wood. Over an impressive 42-year career at Black & Veatch, Ron rose to the role of president and CEO of B&B Energy. And he was lived in Columbia and also St. Louis, earning national respect as a leader in the field of engineering. He remained deeply connected to Mizzou and in 2023 received the Distinguished Service Award for his extraordinary impact on higher education. Mr. Wood was dedicated to an active lifestyle and he encouraged the love of the outdoors, hiking and camping. There he is. And he led a Boy Scout troop for a very long time and took them through the paces, hiking. To support these lectures, they're made possible by a donation from Dewey Shoddy, a former Jefferson Club trustee and a close friend of Ron Wood. So earlier this semester, we heard John Baptiste from Chapel Hill. And today we're going to hear from William Kraus. And I'm going to turn the podium over to Scott, who will do the introduction.
Thanks, Elizabeth. I don't know if the people on Zoom, do I need a mic so they can hear me or are we good? Okay, perfect. Okay, thanks for coming, everybody. Appreciate it. Elizabeth, thank you for inviting Dr. Kraus to speak today. I appreciate that. So I've got to know Bill over the last 6 or 7 years being part of the molecular transducers of Physical Activity Consortium. And it's one of 3 big things that I want to highlight from Bill today. We really are excited about you being here, Bill. I've followed his work since I'll date myself a little bit and then really date someone in the room.
Yeah, boy.
Since the early 90s as an exercise science student at Arkansas State University. He really is truly one of the most influential exercise physiologists that exist in the world. If you think about what he has done and what he has led and how he has made seminal contributions that really have changed the paradigm about what we know about exercise physiology, Dr. Kraus is the guy, right? And so I'll go through a little bit of metrics, and he said, keep it short. I got lots of data to show, right? But he's been at Duke for about 47 years. Before that, he got his degree in astrophysics, his undergrad, and then he came to Duke for medical school, and he's been there ever since. And he's risen up the ranks. He's a distinguished professor of cardiovascular genomics now. Led the cardiac rehab program there for 27 years. Really boots on the ground and the seminal thought leader in this space of one, like what is the exercise prescription that we need in order to have the most benefit? And maybe he's going to share a little bit of sex differences today in that space. But there's really three big studies. The STRIDE study, which was all about modality, exercise intensity. Do we need high intensity? Do we need moderate intensity? And how much do we need? The calorie study, which is part of like the benefits of caloric restriction in individuals that were overweight or obese. He was a leader in that. And then he's been instrumental in the molecular transducers of physical activity. So this is the largest undertaking to map the benefits of exercise across multiple tissues, across the whole human physiology, that included an animal component that my lab has contributed to as well. But those studies are just now starting to come out and under review. And so I think it's really going to change what we think we know that the benefits of exercise. Literally every tissue in the body, all 19 of them we took from animals, are responding with thousands of genes and proteins and transcripts. So, but I mentioned, he really is the seminal guy, the H index of 134, 600 peer-reviewed publications that have been cited over 82,000 times. So top 1% of scientists in the world. And so thankful, Bill, that you came today to share with us. I'm looking forward to your talk. And so let's welcome Dr. Kraus.
What a generous introduction. I don't know who you were introducing though. But that said, my goal, first of all, I heard a number of primes in that introduction, right? So actually cardiac rehab for 31 years, which is also a prime. 27 is not a prime. I'm big into primes. In case you did not understand that. But let's see if I can get oriented here. So my goal is, number one, to have some fun. I always have fun presenting our data. Maybe change some paradigms that you have, or actually develop some new ones that you have in the course of this discussion. And ask questions in the middle. I'd much rather have you ask the questions and we deal with that question than that be in your brain until I get done with the discussion and you've either forgotten the question or because you remember the question, you didn't get anything else out of this, out of the talk, right? So a little bit of pedagogy here. I'm all open to just raise your hand and we'll deal with the questions, okay? All right, great. So a lot of people to thank here, a lot of NIH support, particularly HLBI and NIDDK for the STRIDE studies. And collaborators support from numerous sources. So you don't do work like this without having a lot of collaborators and with a lot of input, both into the science and into the support of the work. All right, so this is my current paradigm of what I'm about in my life, okay? So what we're gonna talk about today is 3 steps, right? What happens? So when you have exercise people, we're gonna talk about insulin sensitivity, but what happens when you put on an exercise training program, right? As Scott said, and what those results look like. So we'll talk first about that. Then the next 2 steps is what I'm really about these days. How does it happen, right? What are the signaling events that go on? What's the communication between tissues, et cetera? And more specifically, if your outcome is insulin sensitivity, what are the proximal molecules, the closest molecules to that outcome in that tissue that could be targeted for potentially repurposing drugs to cause that same effect, right? And that same effect in 3 instances. In people that will not exercise, in people that cannot exercise, and probably as important, people who start exercise and then stop. Can you maintain that benefit, whether they spot for whatever reason, right? Hip fracture, this, that, and the other thing. I think that's useful. I'm focusing on repurposing drugs because we're not about developing drugs. I'm way too beyond that. But there's a whole cadre of drugs at the NIH in a bank that have been developed for a purpose that were abandoned. And that might have the same targets, and we'll talk about a couple of them here, that are identified in the studies that we're doing. So that's the big overview, okay? How does it happen? Our paradigm is this. We have muscle biopsies from tissues. We're focusing today on skeletal muscle insulin sensitivity, okay? What's the input on the training protocol? Our aim one of our competitive renewal is, okay, so aim one, we actually do the molecular phenotyping of the tissues. If I can, young investigators, doing humans, there's one thing I'm going to tell you to do is collect samples and use them, okay? As much as you can get your hands on, because you're going to see the benefits of doing that. Aim two is a global assessment of what's happening with the transcriptome, what's happening with the methylome, what's happening with the proteome. But that doesn't get you to the endpoint. The endpoint is now you start to narrow the candidates with funnels with both a predictive analysis followed by a causal analysis. And I'll talk about what that is here. And then you end up with a circuit diagram or a pathway diagram, or et cetera, where you have your outcome in the middle and you have your most proximal elements. There are other things on the outside, but we want to know what the proximal ones are. What are the proximal signals? And candidate drugs. Then we have candidate drugs. We identify the targets. What are agents available? And we study them in 3D skeletal muscle stimulation models, right? Does that reproduce the molecular patterns that we're seeing? Does it have the effect on insulin sensitivity that we have? So this is our plan. Okay, the three side studies were: one, amount and intensity of aerobic training. Okay, which is more important for which outcome? ATRT, or two, was aerobic and resistance training. Okay, in this study, we had, I'll tell about here in a minute, I won't talk again about this, so I'll talk about it. We studied resistance training alone, aerobic training alone, and the sum of those two exposures. Okay, we didn't downplay the aerobic to account for the time for the resistance. We added them together so that we could ask a mechanistic question. Is when you add them together, is the result the sum of adding them together, or is it more than adding them together? And I'll lead right with the answer on that one. It's more on insulin sensitivity. It's more than adding them together. They're acting synergistically, which means they converge on some common pathway, right? And that's to figure out what that common pathway is. And then the prediabetes study, which I won't talk about either, and that was designed, as I told the students today, to understand a very important clinical question. So we took the diabetes prevention program, which resulted in an improvement in 2-hour glucose and prevention of diabetes, big study, comparing lifestyle to drug, metformin, at the end of the day, to control, where metformin reduced the incidence of diabetes over a 4-year follow-up by 40% and the lifestyle by 78%. And the lifestyle was moderate intensity exercise, 150 minutes per week, plus over 6 months, a 7% weight loss. I'm a clinician. Okay, do prevention, counsel on lifestyle. I cannot ask a patient to lose 7% of their body weight and get this amount of physical activity at the same time. It takes years to get people to do this, right? So I want to know, forget the diet. I'm not a dietitian, et cetera. I'm not good on counseling it. What happens with exercise alone? So we had a clinical group, just like TPP, and they did exactly that, 7% weight loss, about a lot of counseling, all that kind of stuff, and moderate intensity exercise. That was the gold standard. And we said, what exercise will allow you to best achieve that? And it was moderate, high amount, moderate intensity exercise, 80%. Ignore the diet and weight loss, just get them to do that, and you'll get 80% of the outcome, right? Very pragmatic, clinically directed question. Okay, so stride one was this. We asked people to do brisk walking. I'll tell you what the actual prescriptions were in a minute. But brisk walking, 13 miles per week, took about 170 minutes per week. This turned out to be the lower limit of the physical activity guidelines, 150 minutes walking, right? This, jogging 13 miles per week, took 120 minutes per week, is 3 sessions, 40 minutes per week. Does that remind anybody of anything like Frank Booth in here? Those are fitness guidelines that until 1996 were the physical activity guidelines, right? We didn't know anything about moderate intensity exercise. No one even thought about it, right? And then jogging 22 miles per week, 170 minutes per week, we called the Harvard Alumni Study prescription, because this is what resulted in the lowest mortality in the Harvard Alumni Study, okay? I didn't call them this from the beginning. I didn't design it that way. We designed it to what it was. It turns out that's what it is, right? And then we had an inactive group. I used to call it control, but it's not a good control. Controls you don't want to change. These change, and you'll see that in a minute, okay? So these were the actual prescriptions: low, moderate intensity exercise, 40 minutes, 40 to 55% of peak VO2, 1,300 kilocalories per week, energy expenditure, 170 minutes. Low vigorous, 65 to 80% peak VO2, 1,300 kilocalories, 120 minutes per week. And high vigorous, 65 to 80% peak VO2, 2,200 minutes per week, 170 minutes. Okay? So, same amount, different intensities. This is the intensity effect. Same intensity, different amounts. This is the amount effect. You could talk about frequency effect here, because this is like 4 times or 3 times per week, but we didn't design the study to address it. You could only infer what might be the signal, but we can't really answer that question. This was the protocol in stride 1. Brought them in with an assessment. They went 3 months ramp up. This is our first stride study. We're getting people to do vigorous intensity exercise. So these are sedentary individuals. We wanted to minimize musculoskeletal injury risk, et cetera. So we ramped them up for 3 months, and then we did 6 months steady state training at their prescription. We had to adjust halfway through the time per session because they're more fit and they're exercising at a higher intensity, higher energy expenditure, right? So to clamp that energy expenditure, we had to adjust halfway through. Then they went, I won't talk about this today, detraining, okay? But because we have samples from here, we can ask the question, what signals are important for maintaining the effect in muscle, right? With 2 weeks of detraining, right? And then retraining. So in that first stride study, they made us as an optional for the patient. You can't allow that. You can't ask them to detrain for 2 weeks when you train them up for 6 months. That was a criticism. So we added this optional detraining. Half the people took it. All right, fitness, cardiorespiratory fitness. You all know about that. The inactive group will always be in yellow on the left. The low moderate next. The low vigorous. Again, this is intensity effect. This is amount effect. So everybody knows, every athlete knows, the more intense and the more you do, the greater your fitness response. This is what we predicted. This is what happened. So it was kind of a proof of concept. This was triglycerides, however. The inactive group got worse. All the exercise groups, the two on the outside were better than that, but the moderate intensity exercise had the greatest reduction in triglycerides. Interesting. And that was also true for insulin sensitivity response. In fact, this is, again, a comparison of intensity. There's some counter-regulatory thing going on with intensity. Just keep that in your beezer, right? And in fact, you had to do 50% more of vigorous to get the same insulin sensitizing effect, which is reflected in triglycerides, which made us think that's a real biological signal because this is fat insulin sensitivity, right? Okay. However, this was done at a time when women, these are all postmenopausal women, half of them happened to be on hormone replacement therapy. So it allows us to do a natural experiment in this, right? And she said, we know hormone replacement therapy increases insulin sensitivity. That was known at the time. We know it improves HDL cholesterol. It must be good for you. And it wasn't until the Women's Health Initiative came out in the HERS study that we actually discouraged using estrogen. But if you take men and women, the women look like the men. You get the same V-shaped curve, right? However, if you break them out where women are on hormone replacement, off of hormone replacement therapy or on hormone replacement therapy in green, the moderate intensity women look kind of like the men. In fact, the women are outperforming the men on insulin sensitivity. Keep this in your beezer. However, with vigorous intensity exercise, if you are not on hormone replacement therapy, you get no insulin sensitizing effect. There's an interaction between the exercise and the hormone. Exercise pharmacokinetics right here. Pharmacoeffectiveness, if you will. All right, keep that in your beezer. So you got three things to keep, right? Okay, the dose response thing, the women thing, and the hormone replacement thing. Okay, let's go on. Any questions? Are you excited? Yeah, okay, good. All right. All right, I'm going to go back. I know it. All my women friends are, they don't like these data. That's, again, so now we're at how does it happen? Okay, I'm going to get into, I just showed you what happens. How does it happen? So the question is, what are the molecular mechanisms underlying these clinical responses? And then we're going to talk about, can we identify known compounds targeting key nodes in such pathways? And then we're going to talk about, can these serve as clinical mimetics of targeted clinical outcomes with or without exercise? Those are the goals. Okay. Many studies of exercise and insulin sensitivity are only seeing part of the picture. This is why we do systems biology, right? So we can see the whole picture. I won't read all the little bubbles so you can, right? But age, training type, genetics, sex, you know, individual studies, but putting them all together, this is the beauty of MotorPack, right? Is the key here to really understand it, right? So dissecting, I got this from Monica Hubel, the clinical trait, biological variability underlying that trait, physiologic mechanisms, molecular mechanisms, tissue level effects, and then putting it all together. That's our goal. Okay, I think you talked about MotorPack, but just to give everyone, I was telling Vicki today, she'll have some human data in fat available within 9 months that she can download and explore all the B3 adrenergic receptor stuff she wants, right? From the precog data. I mean, this is a credible resource, right, for hypothesis generation. We have 1541 sedentary adults that undergo a randomized clinical intervention, either resistance exercise or aerobic training. And then 10% of the population is control. And then there's a whole collection of healthy, highly active individuals in both those groups as well, across an age span from 18 to 82. Okay, with muscle, fat from the humans, muscle, fat, and blood with proteomics, transcriptomics, um, uh, and in the blood, metabolomics, and in the tissues. And biorepository of samples will be available, and the data sets will be available. And not only that, the code of the MotorPack analysis will be available, right? Okay. That's the MotorPack study design, and here are some references that are already available. In fact, the pre-COG, the before COVID cohort, which is only 206, those papers are posted right now on bioRxiv. So you can get them now from those results. Okay. I'll remind you, molecular mechanisms is the key here. Chris Van has been working with us closely on this, as well as Akshay Bareja here at Duke. I call this integrative global multiomics. Okay, integrative global multiomics is what's the big picture. Okay, so we're going to first talk about epigenetics, right? We're going to talk about DNA methylation responses. So some people believe that a lot of the activity is modifying epigenetics, which modifies gene expression, which modifies protein or activity in some way, the system. Now, it can't be that you have one bout of exercise which demethylates a gene, because then you would only ever have to do forever one bout of exercise. That doesn't work, right? So it must be demethylated and remethylated. And so what's the time course of that? We have a lot of studies of what happens with a single bout of exercise or an exercise training program, but nobody has the detraining data except, right? And we have it at 96 hours and we have it at 14 hours. 96, 4 days and 14 days. And we're just starting to explore that. And of course, as you expect, there are multiple different trajectories in multiple different pathways, right? But what does the global DNA methylation look like? Just pre and post training. I won't go through all that. But mergeomics is the platform on which we're putting the methylation and the transcription together. Okay, it's publicly available. There are multiple sets that you have to go through to do it. There are some recursive modeling in there. I don't have time to go through all that today, but I just wanted to call that out. Okay, effects of exercise amount and intensity on exercise, skeletal muscle methylation, gene expression, and integration is what we're going to talk about here. So methylation is CPG, right? CPG sites on the DNA that get demethylated, but they don't get demethylated as single units. They get demethylated in a region, right? And there's also unwinding of the chromatin to make that place, that area accessible. And there's also modification of transcription by post-transcriptional destruction with microRNAs. Okay, so there are 3 levels of epigenetics here. We're only going to talk about DNA methylation today, right? MotorPack has ATAC-seq, which uses sequencing to identify the methylation sites that way. Okay, but we have been using epic array-based things. So here is low amount, moderate intensity exercise. And these are the significant sites that were at the 0.05 level in the whole methylome that were responsive to low amount moderate intensity exercise. So I'm going to just lay out what the thing is like. These are your volcano plots. So it shows the hypomethylated sites and a negative log p-value here, and the hypermethylated sites. So you get both hypermethylation and hypomethylation. This is 24 hours after the last bout of exercise, okay? And then here are the signaling pathways and the number of differentially expressed positions for each of those pathways. So for example, you see Notch signaling, peroxisome, sphingomyelin metabolism, et cetera, and you pick out your pathway. But I'm really attracted to the chord diagrams because it gives me a big picture of the relationship between these specific CPGs, and the pathways identified here. And it looks pretty orderly.
Of organizing.
No, the mergeomics does this. Wait, just before you make that comment, just hold on. Okay. High moderate intensity exercise. There's a lot more cross signaling on the CPGs to pathways, and they're different pathways. Okay?
Well, is this in the blood or is this in the muscle?
This is muscle. This is all muscle. And this is high amount vigorous intensity exercise, right? These are global. This is not related to insulin sensitivity at all. We'll get to that. But these are global, and it's a different picture. Just a clue, right, from the methylation. Let me show you the gene expression. Okay, I'm putting all the methylation together, and you can see there's a progression here, right? This is this is low amount moderate intensity, high amount moderate intensity. So this is an amount effect, and this is an intensity effect, right? Okay, aerobic exercise, differential methylation, 1/8 of the methylation sites in the 108K thing. High amount moderate intensity shows the greatest number of differentially methylated positions. But vigorous intensity results in a greater ratio of hypomethylated sites, agrees with the current consensus in the literature. And KEGG pathways relate to cell cycle maintenance, metabolism, and inflammatory signaling. Now, why is this slide highlighting high amount moderate intensity? Because this is the STRIDE PD study, and this was the intervention that created the greatest change in 2-hour glucose in that study. It's the one that created 80% of the response of the clinical intervention group, okay? So that's why we're highlighting it here. It's the one most pertinent to the outcome we're interested in. Okay, now how about gene expression? You thought that was something. Look at the low amount moderate intensity. Clean as a whistle. I mean, there are direct relationships between CPGs and pathways with no crossover, right? This CPG is affecting inositol phosphate metabolism, but nothing else. Look at high amount moderate intensity. So now you go to high amount. Oh, there's a lot more action here, right? A lot of crosstalk, right? CPGs crossing over to Wnt signaling and to other pathways. There are multiple connections between CPGs and pathways. And look at that.
This is RNA.
This is RNA, which we, in our modeling, is the most proximal signal. Well, you'll see why. You'll see why. In a mechanistic way. Many more CPGs than the others. I could go back and show you, but there's a lot of crosstalk between pathways here. The response in muscle to high vigorous intensity exercise is complicated, right? It turns out most of this in muscle is driven by women. If you plot out the intensity amount effect, I don't have a slide on this. There's an inverse relationship in women between an intensity effect and an amount effect for a group of CPGs and transcriptions. The men looks flat. They couldn't care. Okay, my paradigm is that women respond to exercise for increasing fitness, molecular fitness, in the muscle. Men respond to intensity. And you'll see this in a minute. Just hold on to that question. Okay? But that's striking. There is no question that the exposures are creating different molecular responses, if nothing else, right? I wanted a dose response in MotorPack. I got outvoted. That's collaborative work. Right? Okay, now I'm going to show you, the, what do they call these diagrams? At any rate, I'll show you what we're showing. These are alluvial plots. They're called alluvial plots. So here's the differentially, this is the DNA methylation marks that you saw before. These are the transcriptome marks that you saw before, and these are the pathways you saw before for low amount, moderate intensity exercise. Okay, what's going on here? Well, between the transcriptome and pathways, there seem to be some islands, if you will, that are pretty uniform, right? And but there's a lot of cross. So the methylation site here is crossing over to these transcriptomes, and then sometimes it's crossing over to up there and down to that pathway. There's some complexity here. And what I really want to point out is there's no one-in-one. We've been searching for this for years, and we're not going to find it, between CPG's demethylation of a gene and transcription. It is not a thing. Don't look for it. You're not going to find it. It's a much more complex biology, right? Maybe you're demethylating a transcription factor that's activating a gene and not a, direct methylation gene thing, okay? It's a program, folks, and it's there for a reason. Okay, here are all three together, okay? Here's methylation gene, KEG pathway. How different do those look? And look at the complexity in the high vigorous. Particularly, you've seen this before with the chord diagrams, right? It's very complex. What does that mean? Right? For understanding how the effects are happening. Yes.
Responders and non-responders in all three of these
Yes.
groups. Does the right side have more non-responders because it's a more complicated system?
Yes, they do. Yeah. I could show you the waterfall plots, right?
Are these the most differentially expressed ones that have plotted?
Yes. Yes.
So then
These are the significant DNA methylation sites. These are the significant.
CO2
Yeah.
response.
Yeah.
And so can I even further and say if you put 100 people on the left-hand side, you're more likely to have more responders. That by going to the right, you're going to uncover
I got to go way back.
non-response because
I got to go way
the
back. Sorry.
systems-
No, I do, because I want to point out something in our science. And then I'll go forward again.
If the response is more complicated, you should get more variance.
Oh, see this here?
Yeah, there it is.
And I show you. All right, I show you these diagrams. You look at the bars. You don't look at the standard deviations. You got to look at the standard deviations.
So it's smaller with the high intensity.
A little bit. It's not the standard deviations, right? But the overlap is, right? Or the distance from the null, right? There are many more people that cross the null here than in this group, right? The waterfall plot looks very different. All right. So bear with me while I go forward, because now we're getting to the exciting stuff. Don't look at it. It's going to make you crazy. But to me, you know, this stuff is just mind-boggling when I first saw this.
So Bill, could you hypothesize what that might look like with 100 participants on a GLP-1 receptor agonist or something? Pick a drug of your choice, metformin. So,
There's going to be a variable response. You'll have non-responders and responders. And the non-responders are the ones that quit the drug or that have a complication. They're getting depression or they're having arguments with their spouse because they're not eating anything. I mean, it's just, yeah. But there's variability in everything, but we ignore it. Okay? We ignore it out of convenience. But if you want to understand mechanism, you can't ignore it. You need to understand what's important and what's not important. Okay. Yeah.
Prevention sensitivity.
Yeah.
Do those include both hyper and hypomethylated?
Yeah. Yeah.
So which
Yeah.
which are hyper, which are hypo? Like the top.
No, they're not separated out that way. Yeah. No, actually they are, right? So I think the hypermethylates are at the top, and then it goes down to the hypomethylates at the bottom. Same thing for the transcription. And that explains the complexity, right? All right, can we identify known compounds targeting key modes? So what we do here is, so Monica's advertisement that she gave at ACSM 2020. These are our causal modelers, Constantine Alferez and CC Ma at University of Minnesota. They have an industry up there of this. I'll talk about what that is in a minute. This is from our first paper that we published last year, NER Molecular Medicine, for this paradigm that we're doing, right? So again, we have the exposure, we have the muscle, we do the gene expression or methylation or metabolomics or whatever we're doing. We have our outcome, and then we look at amount, intensity, and sex-related responses. That's the global thing. And then we develop, in this paper, we developed transcription factors that were differentially expressed. And then we had amount, intensity, and insulin sensitivity related ones, right? And then we put them together and created causal genes in a pathway. All right, so this is what is really intriguing. These are transcription factors related to insulin sensitivity response in our analysis. This is women, this is men. Women respond way more to amount than intensity on transcription factors. And it's almost uniformly repressive. So let's take SP2. They're repressing SP2, the red, where men, intensity is activating it. Men respond to intensity and they activate gene programs. All postmenopausal women.
They're all the same.
Excuse me? 45 to 80, yes.
Young and younger.
All right. So yeah. So look at this this plot here. Okay, this really points it out. All right. So women respond negatively, uniform, except for these two transcription factors. These are the ones that are significantly significant. All right, except for HNF1 alpha and STAT1. Okay. Everything else is repressive by amount. And then they have 1, 2 that are activated by intensity, but they're only 4 in all. Women or men on the other hand, are responding positively to intensity, some of the same ones, right? So here's a woman positively responsive to intensity, as is the men, but men are also replenishing by amount. And there are only two in common. The greater the amount intensity, the high intensity program had the greatest response in these transcription factors in men. And women, they don't care about the intensity. They're responding to amount, right? The more you do of moderate intensity exercise in women, the more repression you get. The more exercise of high intensity exercise you do in your man, you get more activation you get. I told you, I showed you, I said, remember that insulin sensitivity analysis in postmenopausal women on and off hormone replacement therapy. Women not on HRT did not respond to vigorous intensity exercise. This is the explanation. Right? Because none of these people were on hormone replacement therapy. This is stride PD, right? Well, this is stride 1, yeah. And so then predictive and causal modeling. All right, so what the hell is that? So this is their pipeline. It's the best, simplest explanation I can give you. Predictive modeling has a recursive analysis. I'll show you what that is, where you say, okay, how well does this group of genes predict this response? Okay, genes, sex, all that kind of stuff, right? And the recursive, I'll show you again in a minute. There's an inner loop and an outer loop. And they do is, 5, 6, 10 times. They choose a number of different selection tools for winnowing the list. Okay, so we get a list of the responsive genes according to whether it's the tightest signaling pathway that is closest to the outcome, if you will, right? It doesn't go through a different factor. It's direct causal mediator. That's what they conclude from this. If it's in every single model, it is very likely, never say is, very likely that that molecule, that gene is directly causally related to the outcome. Okay, then you can go out another path, 2, you can go out to 2, and then you can go out to 3, right? Then what we do is we put it in the causal analysis pathway, and this is what I was just telling you. You have a Markov boundary. It's called a Markov boundary, and the number of connections to get to the endpoint depends upon which stringency of the model you choose. And then they can tell you, all right, so age isn't in the final model. It's not a proximal mediator. Age got kicked out of the model. What kicked it out? Because it was closer. So you can also select what it kicks out, right? This gene, PPAR gamma is not in the model. Why is PPAR gamma not in the model of insulin sensitivity? Well, it's in the pathway. It's just not in the proximal pathway. Why do I want a proximal pathway? Because I want a drug that is specific for what's going on in that tissue. I don't want any side effects going on if I can avoid it, right? Okay, this is what the recursive modeling looks like. So the inner loop, so you take a group that's the training set, 2/3 is the training set. Then of that, you take 2/3 and you test, and then the test set is 1/3, and then the training set is 2/3, and then the test set. You test this result on this, okay? This is one, by the way, this is one of multiple recursive things they do. They do this like 5 times. Okay, then Then you take a different 2/3 of the population as the training set and then the test set. The middle one is now the test set. And likewise, you do the inner loop multiple times. So you've done the inner loop multiple times, you've done the outer loop multiple times, and you end up with the final predictive equation. And they figure out statistically what the control is. Okay, then we take those candidate genes. We had insulin sensitivity responses from the causal modeling. We had the exercise transcription factors that I showed you, and we have the exercise responsive genes. And then we did the overlap, and these are the 13 items that came out. Okay, and look at that. And look at that. That's TPA. That's the activator of protein kinase. If you give a cell TPA, it will activate the protein kinase. So that implicates protein kinase in the thing, and that implicates also estrogen in the important vectors. Those are the only compounds in this, or these three. But VEGF is in there, TNF is in there, TGF beta is in there, MYC is in there, ESR1. Yep, alpha, that's right. EGFR, I want you to pay attention to this. I published, we took a different data set, STRIDE ED. Jamie Timmons took a double, in the Great Britain, took another HIT study that they did, right, with muscle biopsies and transcription and whatever, and did modeling. And from that paper, different data set from us, EGFR was the highlighted molecule in that drug repurposing paper. So this was validation of that. Okay, so Monica then did, she called these the ringleaders, the influencers, and also the ringleaders. And then she did this in pathway analysis, right? And I just want to highlight this. So here's protein kinase C implicated. Okay, look at all the things it impacts. But I want to highlight that it impacts TGF beta 1, EGFR, and ESR1, right? And beta estradiol. So these are our proximal candidates, but upstream, they're influenced by these. So my paradigm now is, we've known for a long time that activity of muscle, stretching, whatever you want to do, electrical stimulation and culture, rat studies, Lori Goodyear, Eric Richter, all implicated PKC signaling through AMPK, through mTOR, activating insulin signaling. Okay, and it activates the stretching in the membrane or whatever it is, activates the PKC. It comes through AMPK, but so does signaling through the insulin receptor. So the exercise greases that pathway, and then when you're testing the insulin responsiveness, that's where you see the insulin sensitivity response, right? But I would also argue, based upon our clinical result from the STRIDE 1 with the women on and off estrogen, everything that we model, and we've modeled since this paper, ESR1 or beta estradiol comes out. So my paradigm is this. Women, number one, are more responsive. I don't have to show you that. Yeah, I didn't show you that. Women are more responsive to exercise in muscle and men in the central compartment. Probably driven by hormones. Right? So, by the way, just telling you, testosterone gets metabolized to estrone, which is an estrogen signaling molecule. If I have a woman, my wife, who's 3 years younger than I, close them up, my estrone concentration is 3 times hers. It's not like men don't have estrogens around, right? Because we still have some testosterone and that's getting aromatized to estrone, right? Do we want to give men estrogens? Well, I don't know. So but I certainly think women do. So muscle, women, central, men, probably driven by hormones. Women responsive to moderate intensity exercise, men responsive to vigorous intensity exercise in muscle. Okay. Women that don't have it need exogenous estrogens because these are acting synergistically. If my paradigm is right, they're acting synergistically on these targets, right? And that's why women not on estrogen didn't have any response to the vigorous intensity exercise. Isn't that cool? These are our targets. These are what we're testing in vitro now, right? We're going to give to those 3D muscle models. We're going to give them estrogen. We're going to give them an EGFR activator or repressor. And we're going to test TGF beta. But VEGF up here, look at VEGF for all you vascular guys, right? Yes.
Can this be maladaptive and activate EGFR in a cancer promoting type of way though, considering that's a really big target?
That's what the drugs were developed for, cancer, and they didn't work. So I don't know. I mean, it's yet to be known. You're seeing this at the genesis of all of this work, right? But I find it very exciting, keeps me coming to work. You can't do this by yourself. Of course, there's a big team. I want to call out Joe Homard, who is our partner in Stride One. This is our current team. So with that, I'll stop. Yes, ma'am. Of course you did. Do.
But just seeing this even more so, it kind of reiterates the direction that exercise recommendations are going in terms of precision. So I guess, how do you see what you're presenting here guiding precision-based exercise recommendations?
Well, if I were a pre-diabetic or even a diabetic, I don't want them running, walk and walk a lot, right? And that's good message because they don't want to go to the gym and spandex people, and walking's easy. And you know what about walking? Every step counts.
I think moderate intensity exercise is better for women. Should women be avoiding?
Or insulin sensitivity. Let's not get right. If you want to improve your fitness, no. Right? If you want to improve your insulin sensitivity, yes. If you want to increase your HDL cholesterol, that's all about amount. It's not about intensity. Everyone thinks it's all about intensity. It's not. It's about amount. And Larry Durstein published that years ago. But that's escaped everybody's attention. And there are different outcomes, right? We don't know how much vigorous intensity exercise improves bone density. But it's not going to be moderate intensity exercise for sure. So it depends.
I love the plot. The metagenomics and all that data can be hard to visually represent, but I really liked the plot. I think it was really interesting in how it showed the differences that increase intensity and just seeing all of the changes that were occurring.
Yeah. Sometimes you got to look at the big picture to, Yes. Steve.
Like diversity in exercise. And so rather than confine to one level or one duration or one intensity, I like to encourage people to different kinds of activities. For one, alleviate boredom. And so I don't know if you could do that kind of analysis where you've got a very structured protocol for each of the three groups that you're looking at. But in real life, I would like to see diversity in activity. The other thing I wasn't sure about is the age group of the people studying and whether the conclusions that you're drawing are applicable to young, intermediate, and older individuals. So comment on either side.
I think middle age primarily is what we're talking about. In stride 2, which is the resistance aerobic, we went down to 18. Those are really important points, but those are more public health side as opposed to science side. Here we're trying to do the science side, and you got to control your variables unless somebody's willing to give me motor packs type money to do the kinds of things that you were suggesting.
But my bias would be to do as many different kinds of exercises as possible and get all the pathways working. Insulin sensitivity is one that's great, but the other thing is you're sampling muscle, right?
Yeah.
And in muscle, there's more than just myofibers.
Well, I never claimed it was just myofibers.
Right down a list of a bunch of cell types.
Right.
And I don't know if it's possible to do single cell RNA.
We are. Jamie Timmons is helping us do the single cell transcriptomics. So we'll have some signals for you.
That would be important.
Yeah. Yeah.
Not all myonuclei are created equal, as you know.
Right.
And so even within a muscle, you're going to find variability. In muscle fiber, you're going to find variability.
Sure. Sure.
And the other thing, other people here are very interested in is mitochondria. And you ought to be able to, as you probe, figure out what's going on at that level.
Right.
As well.
Right.
So,
It's a quiet crowd.
When are we gonna see the adipose?
Well, I don't have any adipose tissue data for motor pack. The same time all the other data are available. Yeah. Yeah. Well, yeah.
Being a lipid person, what do you think the low intensity, why is that pulling on the lipid string?
Lipids in muscle are not friendly to insulin sensitivity, ceramides, etc. And in fact, I first started getting to the molecular stuff with a postdoc, Irish Smith, that got me interested. And we had 8 genes we were studying, acid ceramidase and what have you. So we know that lipids in muscle are not friendly to insulin sensitivity. When you think about the different intensities, vigorous intensity, they're probably at an RER of 0.9. And moderate intensity, they're probably an RER of 0.78 or 8, which means they're burning more lipid. And so my hypothesis is, and the metabolomics seems to support this, that at the lower intensity exercise, you're clearing the fat, intramuscular fat, you're relieving the negative pressure on the mitochondrial function. And that's why you're getting the more benefit with moderate intensity.
Down in HDL.
Yeah.
Yeah.
Yeah. Okay, well, thank you so much for having me. It was a great day. I really enjoyed myself.